So we finished talking about the cell cycle, mitosis and meiosis, and today we're just going to focus on what exactly happens when we don't control the cell cycle. And that can be extremely dangerous because it can lead to something that we have all heard of before, cancer. Before we dive into understanding exactly what cancer is, let's talk about a little mechanism our cells use to control the cell cycle, as well as control whether or not cells can continue on existing and going through the cell cycle. This is called apoptosis, or programmed cell death. This is a good thing for our cells to be able to do. Apoptosis is a way of doing two things. First of all, regulating cell growth and cell division in case our cells don't divide properly. We don't want to keep making a bunch of copies of cells that are messed up on the inside, so that cell that's messed up will literally self-destruct. The other reason apoptosis is important is for embryological development, and this goes in all organisms. Think about your hands. When you were a developing embryo, you didn't have spaces between your fingers, and as your hands started developing and your cells started dividing and your fingers started getting longer, the skin and the cells between each of your fingers had to stop dividing. Those cells actually went through apoptosis. And the mechanisms by which apoptosis occur, it begins with the chromatin within the cell beginning to shrink. And then eventually the cell membrane will break apart, and that one cell will break into a bunch of tiny little pieces that can then be engulfed by neighboring cells. And we know that process as endocytosis, and it's literally just recycling of all those macromolecules that make up our cells. All right, now that we understand what apoptosis is and what an important role it plays, now we're going to get into what happens when apoptosis doesn't work in a cell and it can't destroy itself when it gets mutated. This is something that's going to lead to cancer, and we've all heard this term before, and you want to know the exact definition. The definition of cancer is it's a disorder where the body loses its ability to control cell growth and cell division. Basically, it can no longer control the cell cycle, and there's lots of different reasons this can happen. If we think back to our cell cycle, we discussed it in the last couple of videos, the cell cycle is literally controlled by proteins, and that's all it is, proteins, and we know what makes our proteins is DNA. And if for whatever reason our DNA gets mutated and we don't make those regulating proteins, such as our cyclin-dependent kinases or cyclins or different growth factors or glycoproteins that work in contact inhibition, we can have some major problems. And basically those cells are just going to keep dividing, going through the cell cycle over and over and over again, and they just will run through those stop signs of those cell signals. So that's what cancer is. Now, one word that we often hear associated with cancer is tumor, and all a tumor is a mass of cells that cannot stop dividing. And it becomes, we know that normally when we have cells dividing and they all perform a common function, we get what we call a tissue, and that tissue performs a function, then it becomes an organ, and so on. Well, when cancer cells are dividing, they aren't like normal cells. They do not have a specific function, and they're very unpredictable, and they just keep growing and growing and growing. And so it's not like if you have a stomach cell that becomes cancerous and it starts dividing over and over and over again, we're going to get more stomach tissue. No, those cells that keep dividing have zero function. They actually will get in the way of the normal function of the tissue that's around it, and this can become very problematic. So what we see over here in the top right, okay, all your beige cells with the purple nuclei are normal cells. One of them started dividing uncontrollably, and that's when it became this blue, irregularly shaped cancer cell. And this mass of cells is what we would call a tumor. Now, sometimes a person's body can get that tumor in check, and the cells around it will release tumor-suppressing proteins that will actually stop this tumor from growing, and then it no longer grows and it doesn't spread. There are two types, like I said, two types of tumor. Our first one are the ones that don't spread, which are benign. And then we have the ones that do spread, and these are very, very dangerous. They're called malignant. And malignant tumors are tumors that have continued to grow. They are not stopping like our benign tumors, and they've grown to the point where they've actually started secreting proteins here. The cancer cells, the tumor cells, have secreted proteins that actually stimulate blood vessels and lymphatic vessels that are nearby to start growing towards it. Because remember, our cancer cells, they're just like our cells. They need a supply of glucose and oxygen. And where do you think they're going to get that glucose and oxygen? they need some blood vessels to bring it over to them. So when this tumor gets big enough, they secrete these proteins, and now all of a sudden we're going to start seeing our blood vessels start growing towards the tumor. So all of a sudden they have their own blood supply. Now there's a couple terms you need to know associated with this. The first term is angiogenesis, which literally means the formation of new blood cells towards new tissue. And this is what happens with tumors. When a tumor becomes so big and they release these proteins, angiogenesis will occur, and that's when a tumor becomes malignant. Because as soon as those blood vessels attach to the tumor, not only do they have a supply of glucose and oxygen, but they can also escape through those blood vessels and travel anywhere else in the body they want. And this is what we call metastasis. Or if we say the tumor has metastasized, that means it has spread. And the only way it can spread is if there are blood vessels and lymphatic vessels attached to the tumor that have now provided an escape route for all those cancerous cells. And like I said before, these cancer cells are very unpredictable. We don't know where they're going to end up, what tissues or organs they're going to attach to. And this is when cancer becomes extremely dangerous. We usually refer to it as stage 3 or 4 of cancer. This is an example of metastasis and how it occurs. You can see over here at number 1 is our original tumor. This is what we would call our primary growth. Angiogenesis has occurred. The red represents our blood vessels. The yellow represents our lymphatic vessels. And for those of you who don't know what our lymphatic vessels are, they're tiny vessels that run right next to our blood vessels, and you can see this picture here is a good example. And they actually carry white blood cells. They also carry the things that our white blood cells break down, and they end at our lymph nodes. And we know that we have our lymph nodes scattered throughout our body. We know that we have some right under our throat, so if you get sick, you can actually feel those lymph nodes start to get swollen, because if you're sick, your white blood cells are going to start dividing, and they're housed in those lymph nodes. But anyways, back to our picture here. We see at number 1, we have our original tumor and the tumor cells over here. Once metastasis or once angiogenesis occurs, blood vessels attach, lymphatic vessels attach. You can see number 2 is denoting where these cancer cells have escaped. Now they can travel throughout the body. And over here, when we get to number 4, we have some blood vessels that have stopped. There's some cancer cells that have attached themselves to this area and will begin developing into what we call a secondary growth. And the other cells will continue moving around the body. And this is when we start having tumors pop up everywhere. And this is very, very difficult to treat. So just a quick review of our 2 major types of tumors. Our benign tumors are what we call localized. They stay put. Angiogenesis has not occurred, so therefore metastasis won't occur. These are the easiest ones to remove surgically. If you catch a tumor and in time and angiogenesis hasn't occurred, we can literally just have a little surgical procedure and scoop it out. If you've ever gone to the dermatologist and maybe they've noticed a little mole on your arm growing awkwardly, they might become a little bit concerned. And so we know that normally if we have a mole, I'll go ahead and do a little picture here. Normally if we have a mole, it should have a nice round shape. That's something that they look for. But let's say that all of a sudden one day your mole starts having some weird edges and it's getting a little bit bigger. And that's what the doctor's concerned with, because basically what's happening here is these cells are dividing uncontrollably, and most likely they're cancerous cells. So all that doctor will do is actually just remove this entire mole. Cut it out in one doctor visit, and that's what you did right here. You just removed this, no angiogenesis occurred, you remove the benign tumor, and you're good. Obviously, skin cancer is the easiest one to catch because it occurs on our skin where we can see it. But imagine these growths inside our body that we don't see. Those are the ones that usually tend to metastasize and spread. And that's when we say we're in stage 2, stage 3, or stage 4, the extremely dangerous form of cancer. And this is when we have a malignant tumor. Okay, the malignant tumors are tumors that have metastasized because blood vessels have attached, and now these cancerous cells are spreading throughout the body. So what exactly causes cancer, and what causes it to happen? What causes these cells to go out of control in cell division? Well, there's a couple terms we need to know to begin with. First, we have to understand what a mutation is. And we've talked about DNA and nucleic acids, and we know that a gene is a section of DNA that codes for a specific protein. And so when we talk about a mutation, it's literally saying we change that section of DNA so we no longer make that specific protein. Now, there are mutations that can be beneficial, and there's mutations that can have no effect whatsoever, and then there are those very, very dangerous mutations. Now, usually when cells undergo some sort of mutation, because of apoptosis, they're able to self-destruct so that they don't divide and become 2 or 4 or 8 cells with that mutation. Our white blood cells will also come and detect a cell that has undergone a mutation. Now, cancer cells are unique because they are mutated, but they can bypass all these fates of mutated cells. Cancer cells will not go through apoptosis, and the white blood cells cannot destroy these cancer cells. The next term we need to know is mutagen. Mutagen is anything that can cause a mutation, and we already know about some of them. UV light is a mutagen that causes mutation in our skin cells, and that's what can potentially cause our skin cells to become cancerous. If you wanted to take it to the extreme level, the radioactive spider that bit Peter Parker can be considered a mutagen, because when he bit Peter Parker, he actually changed Peter Parker's DNA to the spider DNA, and then you got Spider-Man. But anything that causes a mutation is considered a mutagen. Now, a carcinogen is anything that causes cancer. Now, not all mutations cause cancer, only some of them do. So this is important to understand the difference. Some mutagens are carcinogenic. And that's an important term to know. And we've heard of carcinogens, and in lots of tobacco commercials you hear them talking about how cigarettes are full of carcinogens, and all that means is they're full of cancer-causing agents. And those cancer-causing agents are mutagens that destroy or affect your DNA in a negative way so that they no longer make the proteins that control the cell cycle. And finally, there's another term in which we call oncogene. And oncogenes are referring to a section of DNA that has been mutated and now will end up causing the cell to become cancerous. And if we go ahead and look up on this picture in the top right-hand corner, okay, proto-oncogene, all that means is it's a gene before it's been mutated and before it becomes an oncogene. There's some sort of energy radiation that comes in this case UV light, chemicals, etc., okay, that will break the hydrogen bonds in this section of DNA, and all of a sudden this normal gene becomes what we call an oncogene. And now that cell is cancerous, and it will begin dividing uncontrollably. It cannot go through apoptosis, and it will eventually become a tumor. Now, what exactly will cause a mutation to our DNA that will cause a cell to become cancerous? Well, there's 4 major causes or factors that increase the mutation rates of our DNA. And you do want to know these 4. Some are more common than others. First of all, we have ionizing radiation. This is any type of radiation, whether it's UV light, X-ray radiation, gamma radiation, nuclear radiation, that will penetrate the nucleus of your cells. It will actually get into the DNA and break those very, very weak hydrogen bonds. And now that if they happen to be the hydrogen bonds in the section of DNA that codes for a protein that is important in the cell cycle, then that cell will become cancerous. The second cause of mutations is going to be chemicals. And this is when we hear that term carcinogens. There's lots of different carcinogens out there. The ones in cigarettes is just one type. We also have carcinogens such as asbestos. You may have heard of asbestos in building materials. Is actually in the insulation of very, very old buildings. And they started realizing that people who lived in these buildings or worked in these buildings had higher incidences of lung cancer. And they determined that asbestos was a carcinogen, was a chemical that caused lung cancer. And that's all because when someone breathes in this chemical, it reacts inside of their lungs and releases just enough energy to break the hydrogen bonds in the DNA of their cells and cause them to become cancerous. Next, we have viral infections. And if you know anything about viruses, viruses carry their own genetic material, sometimes DNA, sometimes RNA. And what these viruses do is they need a host cell. They're not living on their own. They're basically a protein coat. If we look over here, this is kind of what a virus would look like. It's a protein coat, and that's what would be in red. and they need a host cell, and inside of that protein coat, what they have is their DNA. And so we come over here and we'll put some viral DNA here. And what this virus wants to do in order to reproduce, it needs to inject a cell with its DNA. So it will find one of your cells, inject its DNA, and then all of a sudden, whatever that DNA says is what that cell will do. And if it happens to be carrying an oncogene, that means your cell will now become cancerous. A very common viral infection that causes cancer is a human papilloma virus or the HPV virus. And right now we have a vaccine for it, so you can get vaccinated so it can prevent the spread of this virus. But it does lead to cervical cancer, and it can be extremely dangerous. Now, the fourth cause of cancer, origin of cancer that we know about, is genetic predisposition or your hereditary disposition. And all this means is that we do have some forms of cancer that are genetic. One of the most common forms of genetic cancer is breast cancer, and that's because there are two genes that can end up being mutated and passed off to offspring, and if the offspring inherit this gene, they're going to be at a higher risk for contracting cancer throughout their life, and we'll go into that in just a little bit. What's important to remember is that when we talk about how these four factors affect or cause cancer, it all has to do with DNA and the proteins that are made because of the instructions in DNA. We know that proteins regulate our cell cycle. We know that these proteins are made because of specific sequences on the DNA, those A's, T's, G's, and C's. And those sequences of DNA are held together and kept in the correct order by hydrogen bonds. And if enough energy gets in there to disrupt or break those hydrogen bonds, then if it happens to be the section of DNA that codes for those proteins that are regulating the cell cycle, then we will no longer make those proteins. And then if we don't make those proteins that regulate the cell cycle, the cell will just keep dividing uncontrollably. The technology we have now, scientists have been able to actually look into our DNA and determine what genes, if mutated, will lead to cancer. And one of those genes is the p53 gene. Now, all you have to know is the p53 gene is just a tiny section of DNA, and it actually codes for proteins that if a cell becomes mutated, it will release these proteins to stop the cell cycle. And that's a really important job. It's a very, very important protein, these p53 proteins made by the p53 gene. Now, if the p53 gene happens to be mutated or it's defective because of excess radiation, that is a big problem, because if it's mutated, it will no longer be able to make those proteins that stop the cell cycle from dividing. And if a cell is mutated, that cell will divide uncontrollably until eventually it becomes a tumor. So what you see over here is a cell that has a p53 gene, and if it gets mutated and that p53 gene works, it repairs the mutation. If it can't repair the mutation, it destroys itself. On the bottom, though, is an arrow showing if this cell does not have a p53 gene, a mutation occurs, it divides uncontrollably until we have hundreds and hundreds of cancerous cells that have the potential to spread. The second pair of genes that scientists have directly linked to cancer are called the BRCA genes, which the BR stands for breast and the CA stands for cancer genes. Now, we all have BRCA genes. They're actually located over here on chromosome 13 and chromosome 17. And these are also tumor suppressor genes, meaning these genes are going to code for proteins that stop the formation of tumors and prevent cells from becoming cancerous and spreading. We have two of them, BRCA1 and BRCA2. They actually repair damaged DNA. If the damaged DNA cannot be repaired, it does signal for apoptosis. This ensures the stability of the genetic code, the stability of the DNA, and makes sure that we're only replicating DNA that is properly replicated. However, If a BRCA gene becomes defective or it becomes mutated, it can be extremely dangerous because that means those chromosomes are no longer able to code for a protein that stops the tumors from spreading. Now, a person can have defective BRCA genes their whole entire life and never get cancer because those cells never became cancers. However, if they don't have if they have a defective BRCA gene or a mutated BRCA gene and there's UV radiation or energy or something that causes a cell to become cancerous and start dividing, then they can't stop that cell from dividing. So what's important to understand is that we do all have these BRCA genes. However, they can be mutated and these mutations can be passed off from one generation to the next. That's why they say it's genetic, or it can be genetic. And just because you have it doesn't mean that you're necessarily going to contract cancer. All that means that if a cell does happen to become cancerous at some point in your life, you won't be able to stop it from dividing, and then that leads to cancer. Now, how exactly do we treat cancer? There's 3 major ways we can treat cancer, and these are very common, and I'm sure you've heard of them. The first way we can treat cancer was with surgery, and we mentioned this before. If you catch a tumor before it's become malignant, if it's still a benign tumor, it has not metastasized, surgical removal of the tumor is possible, and hopefully when you do have that surgical removal, you are able to take out all of those cancerous cells. This can really only be used for localized tumors or tumors on certain tissues that if we remove part of that tissue, it's not going to be harmful for that person. Melanomas is a form of skin cancer, as I mentioned before, and it's very common to have a skin section, a piece of your skin or section of your skin or a mole or freckle start dividing uncontrollably and become cancerous. And if you catch it on time, if you're constantly checking your skin, looking for any abnormalities, then you can prevent this from spreading by getting it removed at the doctor's. The second form of treatment is radiation, and we know already that radiation is excess energy. We know that it can break or disrupt the hydrogen bonds holding together DNA. So if we use radiation on an area where there is a tumor, we can actually disrupt all of the DNA in those cancer cells. And if those cancer cells don't have working DNA, they can't divide. So that works, but one of the problems with that is that we also kill some of the cells, some of the healthy cells or normal cells around it. And this picture here is giving you an idea. Here's our tumor. Those are all the purple cells, and then we see our normal healthy cells in pink. When a radiation treatment begins, all of the cells become damaged, including the cancer cells, and we see the dying cancer cells are in gray, but we also have some damaged normal cells. And with continuous treatment, we begin shrinking the tumor, more of the cancerous cells die until eventually the normal cells can replace themselves and we've gotten rid of it. Again, this has to be in the preliminary stages of cancer before metastasis has occurred. The final treatment, I think, is the most common that we've heard of, which is chemotherapy. And all chemotherapy is a mixture of medications that a person is going to take through an IV. And these medications actually target all dividing cells. And when I say target all dividing cells, that means those cancer cells that are dividing uncontrollably, going through mitosis over and over and over again, those will be directly affected. In addition to any other cell in your body that's dividing, this is going to include all the cells of the digestive system. Those cells divide every few days, so those cells will no longer be able to divide. It also includes the hair follicle cells. That's why oftentimes when we have friends or family going through chemotherapy, they do lose their hair because their hair follicle cells are no longer dividing and they can't grow back their hair. And then we also have our white blood cells. Our white blood cells, when you get sick, they go through many stages of mitosis in order to build up an army to fight off that infection. A person who's going through chemotherapy, if they get a common cold, they won't be able to have their white blood cells divide, and that could be a big problem. So chemotherapy directly targets cells going through the cell cycle. It actually prevents different stages of mitosis from occurring. Sometimes they prevent DNA from replicating during the S phase. Sometimes they actually prevent the spindles from attaching to the centromeres during metaphase. So therefore, those sister chromatids can never split up and the cell can never divide. So chemotherapy is just a set of medications that target any dividing cell in the body, including those cancerous cells. Hopefully, after many rounds of chemotherapy, all the cancer cells have been destroyed, and that's when we say a person has gone into remission.